Hello students, welcome to EPG Pathsala. I am Professor D. Velmurugan, former head of the department from Center of Advanced Study in Crystallography and Biophysics, University of Madras, Gindi Campus, Chennai, and I am at present working there as the University Grants Commission Basic Scientific Research Faculty. Today, we are going to talk on the module, some of the macromolecular structure determination programs, CCP4, PHENIX suite, SHELX-D and SHELX-E from paper Biocrystallography. Let us first consider what are the objectives we are going to deal with. First, I am going to introduce you to the suite of programs called CCP4, Computation Collaborative Project number 4. And we are going to look into some of the programs in the list. The program is a list contains a lot of programs. And some important programs I am going to tell you. Then, as an example, we are going to talk about two suites of programs. One is the PHENIX suite, another is the program SHELX-D and SHELX-E developed by the German physicist Professor Georg Sheldrick. Macromolecular structure determination is carried out by many modules which are embedded into two major softwares, namely CCP4 and PHENIX. The SHELX module is used for substructure identification, what we mean by location of heavy atoms, and also for model building. The presentation puts forward the different modules available for structure determination from initial phase identification to the final validation of the modeled structure. The name of the module and its function has also been provided. CCP4. What do you mean by CCP4? It means Collaborative Computation Project number 4. And it's a free software. Anybody can download them into the computer and use it. It's user friendly. And this is a program suite which is an integration of a set of programs in protein crystallography written by many scientists, and which includes a large number of very useful tools developed by these scientists around the world. The programs which are available as supporting modules in CCP4 are listed below with the name and the purpose for which each program is useful. Extensive details can be found about the program suite from the following reference and website: Collaborative Computation Project number 4, 1994, the CCP4 suite, Programs for Protein Crystallography, which was published in Acta Crystallographica section D 50, page 760 to 763. The website is http://www.ccp4.ac.uk/dis/html/index.html. We have earlier seen that CCP4 is nothing but the Collaborative Computational Project number 4. It is a suite of programs, and these programs have been written by many scientists. Present day CCP4 version is very simple and user friendly. To illustrate that, we are now just showing some graphical interface of CCP4. So if you look at the left hand side arrow mark column, we have the various programs listed there, and in the right hand side working directory is given. For example, let us take the macromolecular refinement program called RefMac. So this is the interface for RefMac program. Like that, whatever the program you are going to execute, which you are going to detail later, so we have these windows appearing and you can enter the input. So this shows the execution of the program SHELX-C, SHELX-D and SHELX-E, which is a pipeline interface. So we are going to give the input in MTZ format, where we have intensities and structure factors provided in the input.mtz. Then output files name, everything we given, which will be generated in that name you are given. So this lists the programs contained in CCP4 along with their purpose. As time is short, I won't be able to explain all the programs, and some of the important programs I will try to explain you. The first program, there's a program called ABS, which will determine the absolute configuration of the heavy atom substructure. And the ACON means it's a program written for application of direct methods in case of macromolecules when you have the atomic resolution data available, and this program comes from the University of York group from England, and this has molecular replacement, Sayer equation and dynamic density modification for the determination of a protein structure. This program ACON will be dealt in a separate module. And we take other programs like analysis of coordinates, which is called ACT, and scaling of intensity aimless, and AMORE is an important program for doing automated molecular replacement. This abbreviation called MORE, and this has been written by George Navaza from France, and it's an automated molecular replacement program. The earlier version of this automatic molecular, before this automation came, so one used to do molecular replacement using a program called MERLAT written by Professor Paula Fitzgerald. So then we have anisotropic displacement parameter analysis for the program ANISO-ANL and so on. Then for solvent correction we have a program called bulk. Suppose you have the crystallographic reflection data from several files, how to collect and sort. So for that CAD is used. And we have other program called chainsaw to mutate a PDB file according to an input sequence alignment. So we have the program called Cluster W interface for the multiple sequence alignment. No, this is a graphical interface program. And the multiple isomers replacement density search, we have the program called CMAP cut. We have to do the composite omit maps program called commit. Then the conversion to one format to another format, we have convert to MTZ, from CNS to MTZ format, and to produce the MTZ file from the input file called Scala, we use combat program. Then automated structure solution for experimental phasing, we use a program called crank. So we have a program called DIMPLE for automated difference map calculation. For calculation of distance and angles, we have program DISTANG. Density modification, again we are going to deal about this in depth. So it's a program called DM, and to determine the dynamic domains when two conformations are available, we use the program DINDOM. We have a program to calculate the normalized structure factors. This is known as eCalc. To calculate electron density maps, one uses ELSTATS. For the fast Fourier transform, you do the program FFT, and map interpretation package, you use FFR, and so on. Molecular geometry calculation, we use GeomCalc. Structure alignment, you use AMT. Then to display the zones of reciprocal spaces as pseudo precision images, we use HKL view. Suppose you want to view as a plot the precision photo from an HKL data file, we have program HKL plot. If you want to generate hydrogen atom position for proteins, we have the program HGen, and so on. To view the CCP4 format log files, we have the log graph. Then to convert CCP4 map to crystal view format, we have program map to FS. Then to do this map or mask extend program, you use map mask. And to convert binary map file to and from NA4 ASCII format, we have the program mapton NA4. We have an important program called MLFAIR, which is called the Maximum Likelihood Heavy Atom Refinement and Phase Calculation Program. As I told you, another important program is MOLREP, which is an automated program for molecular replacement. So for processing the image plate and CCD data, one uses MOSFILM, MOSFLM version. Then for depositing your structure factors, we do use the MTZ to shift. Then for reflection data file utility program, we use MTZ utils. We have a program called NPO for molecule and map plotting. We have the program for breaking the phase ambiguity in one wavelength anomalous scattering or single isomorphic replacement. The program is Y-axis. This is written by Chinese group. We have to read in the reverse way. Y-axis means Institute of Chinese Academy of Sciences. So this is called Y-axis, Academica Sinica, Chinese Academy of Sciences. Then we have a program called OMIT. So this is developed by Indian scientist Dr. Bhatt, who worked with Professor Blow, and they developed a program of OMIT maps. So to cross-check whether you got the amino acid sites at proper position, it's good to omit certain sequences and do this map. If the true phases are for other residues are genuine and reliable, automatically the omitted residues will come at proper positions. So this we can do at various segments by omitting those segments and getting it back to confirm the thing. Again, we have a program for density modification package called Parrot, and we have the important program called Phaser for doing a molecular replacement, single wavelength anomalous diffraction phasing, anisotropic correction, cell content analysis, translation, non-crystallographic symmetry, and twin analysis. It's a very big and important program. Then to determine the Lavie group, we have a program called Pointless. For fast rotation function that works in polar angle, we have a program called Polar RFN. We have an important program called ProCheck. So these are the programs to check the stereochemical quality of protein structures you are predicting on the basis of your structure determination. Then we talk about program Rampage. This is important thing. Before you propose the accurate model, you have to validate it, and you have to prove that the torsion angles phi and psi occur within the allowed region proposed by Professor Ramachandran. When Ramachandran proposed this plot, that time only few protein structures are available, one or two protein structures are available. This is entirely based on the peptide structures, small molecule structures. So based on which he developed like a figure containing the contours, showing the allowed region of the phi and psi, which has now become a validity check for the protein structure determination. They want to honor Professor J. N. Ramachandran because he has contributed voluminous amount of research publication towards the confirmation analysis of polypeptides and proteins. Then we have a program called RANTON, which is a direct method module for determination of heavy atom positions, and we have the famous protein refinement program FMAC5 for doing the macromolecular refinement. We have a rotamer program for listing amino acids whose side chain torsion angle deviate from the rotamer library. We have a program called RSPS for the heavy atom position from derivative difference patterns and maps. And if you have two sets of structure factors, how to scale together. There's a program called RSTATS. And to analyze the shape complementarity, we have a program SC. For heavy atom site location, there's a program from China is used SAPI, that is the reverse way, Institute of Physics Academica Sinica, the Department of Physics, Chinese Academy of Sciences, that you have to read. Then for scaling together multiple observation or reflection, you use Scala. We have programs for statistical model building called Sequence, and for checking the agreement between the atomic model and X-ray data, we have SF check. For the improved Fourier coefficient using calculated phases, we have program Sigma. Then another important program is for density modification by solvent flipping. This is known as Solomon. Then for exact coordinates from stereo diagrams, we have the program Stereo. So we have a program called Superpose for structural alignment based on secondary structure matching. We have TFFC for the translation function Fourier coefficients. Then we have a topological comparison program called TOP, etc. To generate patterns and vectors from atomic position, we have vectors program. And for doing Wilson plot to determine the absolute scale and temperature factor, we have the program Wilson. To select peaks from peak max and to put them close to respective protein atoms, we have the program Watt peak. And to rationalize waters at the end of the refinement, we have the program Water tidy. Let us now consider the program suite PHENIX. PHENIX is a comprehensive Python-based system for macromolecular structure solution developed mainly by Paul Adams group, and Tom Terwilliger has a major role in it. Publications deal about the PHENIX. So one reference I have given. During the macromolecular structure determination, one may have to use many software packages and repeatedly use interactive three-dimensional graphics. A comprehensive system for the above problem with automation of all procedures has been developed as the PHENIX suite. The scripting layer is the heart of the system. With only a few exceptions, all major functionality is implemented as modules that are exclusively accessed via the scripting interfaces. The object-oriented Python scripting language, reference here, is used for this purpose. C++ is used as the language. The current PHENIX release includes graphical user interfaces GUI for PHENIX, Refine, PHENIX.XTH, AutoSolve, AutoBuild, and Ligand Fit, wizards, the restraint editor reel, all of the validation tools, and several utilities for creating and manipulating maps and reflection files. Interface for automolecular replacement AutoMR wizard and Phaser are also now available. Let us analyze the above programs in a detailed manner. PHENIX has a range of tools for the analysis, validation, and manipulation of X-ray diffraction data. PHENIX.XTH is a comprehensive tool for analyzing X-ray diffraction data. This carries out tests starting from space group determination and detection of twinning to detection of anomalous signal. We have seen what for one uses the PHENIX program. Again, this is a recent version of the PHENIX. It's user-friendly. They have developed the graphical user interface called GUI for executing PHENIX. That is shown in this slide. PHENIX.XTI range provides the user with feedback on the overall characteristics of the data. Database derived from standard Wilson plots for proteins and nucleic acids are used to detect the anomalies in the mean intensity. Data strength and low resolution completeness are also analyzed. Measurability of statistically significant bifid differences in a data set is used to check the presence of anomalous signal. So this problem is one of the statistical applications in crystallography developed by my mentor Professor S. Parthasarathy from Crystallography and Biophysics Department, University of Madras, who was a student of Professor G. N. Ramachandran, and one Dr. Peter made use of this publication and developed this program. After ensuring that the diffraction data are sound and understood, the next critical necessity for solving a structure is the determination of phases. For finding a substructure, we have a program called PHENIX.HYSS, called HIS, hybrid substructure, which combines multi-trial dual space recycling procedures, pioneered by Shake and Bake, written by Miller and et al. So I was also a part of this program in the initial stages when I worked with the Nobel laureate Professor Herbert Hoffman at Medical Foundation of Buffalo, New York. Shake and Bake is a program where a minimal function is used. A set of functions is defined which will take minimum value when the phases correspond to true values. What do you mean by dual space refinement? We have real space in which the density is modified, and we have reciprocal phase where the phases are refined. So now SHELX-D was released by Sheldrick for the use of first translation function. For phasing, as we mentioned earlier in the CCP4, phaser program is available in the PHENIX.phaser, where the principle of maximum likelihood is applied to solve crystal structure by molecular replacement, single wavelength anomalous diffraction, or by a combination of both. Non-crystallographic symmetry NCS is an important feature of many macromolecular crystals that can be used to greatly improve electron density maps. PHENIX.find_NCS and PHENIX.simple_NCS_from_PDB are tools for the identification of non-crystallographic symmetry in a structure using information from a heavy atom substructure or an atomic model. For building an initial core model and ligand fitting, For the rapid model building of secondary structure and main chain tracing, PHENIX.find_helices_strands is used. For the fitting of flexible ligands as well as for fitting a set of ligands in a map, PHENIX.ligand_identification is used, and PHENIX.fit_loops is used for the fitting of missing loops. So thus, PHENIX can be used for main chain tracing and also for fitting a set of ligands and also for fitting the missing loops. Validation tools are also provided so that the models produced can be validated at each step along the way. So numerous research publications have been made by Professor Tom Terwilliger and also by Paul Adams, the two principal authors of this suite called PHENIX. So we have Resolve, which does the model building. So this is a part of a PHENIX suite. PHENIX.refine and the graphical tool PHENIX.create_maps can produce various types of maps including anomalous, difference, maximum likelihood, weighted and regular maps. A computationally intensive but powerful method of creating a very low bias map is to carry out iterative model building and refinement while omitting one region of the map from all calculation of structure factors. This has been done by the publication by Terwilliger in 2008. The PHENIX.autobuild iterative.build omit map procedure carries this out automatically for either a simple omit regions or for overlapping omit region to create a composite iterative_build omit map. PHENIX suite includes many individual tools for specific aspects of validation plus several systems that combine those results into overall summaries. Most aspects of model probability, model validation tools have been adapted for integral use with PHENIX. Hydrogen atoms are added by PHENIX.reduce. PHENIX.validation_model analyzes the deviations of intermolecular features. PHENIX.real_space_correlation provides a quantitative measure of how the atomic model fits the electron density map at the residue or atom level. PHENIX.validation combines all of the tools in one GUI, providing a single place for assessing the results of structure determination. Automation has dramatically changed macromolecular crystallography over the past decade, and every week you see around 125 to 150 structures are deposited. This is not only due to the synchrotron radiation development or the SAT technique, but also due to mainly the automation in the structure determination also. The great speeding up the process of structure solution, model building and refinement, and by bridging the tools for structure determination to a much wider group of scientists, they allow the determination of even more complex and difficult structures possible. Let us now learn something about the SHELX suite of programs. Multiwavelength anomalous diffraction MAD method has been greatly benefited by the recent advances in synchrotron technology, cryocrystallography, and the incorporation of selenomethionine into proteins, and has become useful and effective approach to the structure determination of macromolecules. The other powerful alternatives are single wavelength anomalous diffraction SAT technique with native anomalous scatterers like sulfur or phosphorus, or soaking experiment with the halides or iodine combined with multi-wavelength anomalous diffraction or single wavelength anomalous diffraction experiments. In the above methods, calculation of phases from these heavy atom sites is more important. Although Patterson maps are very much useful in the above calculations, direct methods have also been proposed. One knows that for the direct methods to work, the data had to be at atomic resolution. Only at atomic resolution the peaks are well resolved. There is no overlapping of electron density peaks. But as far as the solution of the heavy atom substructure is concerned, the resolution of the data, say 3 angstrom, is smaller than the separation of heavy atoms. Here, dual space direct methods will be very useful in the situations. Shake and Bake program from Hoffman's group uses dual space recycling approach, which alternates between real and reciprocal space. As I have indicated earlier, Professor Hoffman has developed a function called minimal function based on the cosine value of the triplet and its expected value. theoretical expected value, and the difference has to be minimum. He has made use of weighting factor. I was very much involved in the verification of the minimal principle. In the case of small molecules, we have done up to isoleucinomycin, a polypeptide, and we proved the working of the minimal principle when Hoffman formulated this. The values of the cosine of the triplet phasm is minimized, as I told you in the beginning, with respect to the expected value given in 19.2, which is the ratio of the Bessel function, modified Bessel function of order 1 to order 0. The argument of the Bessel function is A, which is like in the small molecule case, it is proportional to 2 by square root of n, the modulus of the 3 E values, products. So here, as I told you, the probable estimate comes from a reliable probability distribution where the variance has to be very small. If you look at the A value, when n increases, the A value becomes small. Whenever A value becomes small, the variance becomes larger. So wider distribution is there, so probability becomes very small. So that is why direct methods fail in the case of proteins. But by using this minimal principle, Hoffman converted this problem into a multi-parameter problem where multiple minima are there from which unique minimum can be found corresponding to the true phases. So it's a minimization problem. SHELX-D uses real space recycling approach. 1/3 of the peaks are left out at random when phase values are calculated. Here, both the electron density modification is done in real space and phase value refinement is done in the reciprocal space. In cases where the deleted peaks are the correct ones, they will reappear again in the Fourier after the reciprocal space phase refinement. For improving the efficiency of the dual space algorithm, the Patterson peaks can be used in the start instead of random starting atoms. SHELX-D uses unsharpened Patterson for substructure solution. Substructure means determining heavy atoms. For large number of substructure atoms, calculation of a full symmetry Patterson superposition minimum function PSMF is useful. Peaks are obtained by map interpretation and sorted. This procedure is applied before each run through the dual space recycling, and it is possible to generate an unlimited number of different sets of starting atoms. SHELX-D identifies potential solutions by the high values of correlation coefficients CC. For ab initio applications, these potential solutions can be improved and extended by means of peak list optimization. With moderate structures with good anomalous signals, SHELX-D works efficiently fast. SHELX-D works with single wavelength anomalous diffraction data as well as multi-wavelength anomalous diffraction data. Here, the CCs can vary from 40 to 75% depending on the size of the structure. Although SHELX-76, the program released by George Sheldrick, the German scientist, for the refinement, was introduced as the first package. The more sophisticated procedures and use of negative quarters to eliminate false solutions were later implemented, leading to the various SHELX 86, 90, 93, 97. These are various years in which the different versions of the program was released for solving small molecule structures with up to 100 unique non-hydrogen atoms. Simulated annealing approach was used in SHELX 90. Although many direct methods programs like shake and bake, semi-invariant representation program written by Professor Giacovazzo's group from Italy, and SHELX-D are available, SHELX-S is still widely used. For sharpened Patterson, F square was replaced by square root of E cube F in SHELX 86 and SHELX 97. In SHELX 86 and the later versions, iterative EMF recycling facility is available to expand from incomplete direct methods or Patterson solution to the full structure. Rubredoxin was used as the initial test in finding FeSO4 unit and cramine in finding the 6 sulfur atoms by automatic Patterson interpretation. These structures were later expanded to their full structures also. Detailed publications are available. SHELX-C, SHELX-D and SHELX-E, all these programs which come out from Professor Sheldrick lab, are useful for experimental phasing for macromolecules. Heavy atom sites can be located from SAD, SIR, single SMM replacement combined with anomalous scattering, or MAD or RIP, radiation induced phasing data from macromolecules. Once heavy atoms are found by SHELX-D, experimental phasing can be carried out by the program called SHELX-E phase extension and refinement. The heavy atom phases are used in SHELX-E to obtain starting native phases, which are later improved by density modification. When the direct method program Econ for macromolecules was released in 2001, the idea that if the solution of the native data is almost 2 angstrom or better, it may be possible to improve the density and phases by extrapolating to higher resolution than was actually measured. We did some modifications on that while feeding the input and made it to work even at a resolution lower than 2 angstrom, and having got encouraged by this, Fodi, the main author of this program, Econ, appreciated our attempt, and the later versions that's also been incorporated. Now the program can work automatically up to 2.5 angstrom or so. So now this Econ is also now added to SHELX-E program using sphere of influence algorithm. Phases are calculated by density modification. The extrapolated amplitudes are obtained from Fourier transformation of the density modified map and are then normalized to fit the extrapolated Wilson plot. SHELX-C prepares the 3 files necessary for running SHELX-D and SHELX-E. SHELX-D and SHELX-E have facilities for rip phasing also, along with calculation of anomalous Fourier maps. SHELX-L can be applied to macromolecular refinement also, like your RefMac. Conjugate gradient refinement, the free R factor and non-crystallographic symmetry restraints were added in this program to facilitate the protein refinement. SHELX Pro is distributed along with SHELX-C. SHELX has been able to maintain its dominant position in both small and macromolecular crystallography due to its frequent updates incorporating the latest developments in the field. So this just shows the SHELX-C output, just to have a feel of the run of SHELX-C. SHELX-D output, where like a small molecules, cards denote title, cell giving the first value as a wavelength, then ABC, alpha, beta, gamma, lattice, negative sign shows non-center symmetry, 1 means primitive lattice, then symmetry positions are given in the 3 rows, then scattering factor is given as selenium atoms, so they are given 128 selenium atoms. We just see within minutes it has found selenium atom positions. So you have in the third item along every row, starting from X, then Y, then Z coordinates are there, then occupancy of the selenium atom is given. So these are all called heavy atom positions, which are output by SHELX-D program. So this is just to have a feel of the map produced by SHELX-E, to have a comparison with the direct methods program map y-axis and the conventional, the difference Fourier map 2FO minus FC, which is provided as a third map. A feel you can have now for the 1.7 angstrom data. All the 3 procedures work very well. There is a direct methods phasing map, y-axis map, first map, second is SHELX-E map, third is your final difference Fourier. So the first 2 maps match with the actual 2FO minus FC map, validating the efficiency of y-axis and SHELX programs. This shows the electron density map of actidine at 1.5 angstrom. This shows how clearly the map will look like when you have this atomic resolution. At each peak we have a bulge of the electron density. So students, let us summarize what we have learned in this module so far. The present module throws light on different softwares and their uses for phase identification, model building, density improvement and structure validations. We looked into the suite of program first called CCP4, which contain a lot of programs written by many scientists all over the world. Each program has its own usage, and there were many programs meant for a common use. So it is all user friendly, and as I've been repeatedly telling in the presentation, don't be stuck to a single program. If a single program doesn't work, use another program which has been meant for the same purpose. And also we have seen 2 other packages. One is the PHENIX suite of programs. In PHENIX, there are two modules, solve and resolve. Solve uses the heavy atom positions located from the anomalous scattering data, and resolve uses them for phasing and doing your modeling business. So it is separately available, and also it is contained in the PHENIX suite, which also has some other programs other than these modules. The names of the software and its function have also been provided with its role at different stages of such a solution, starting from data reduction and applying the thermal factor corrections, calculation of normalized E structure, E values, then rotation function, translation function, the molecular replacement techniques by various programs, then your starting phases from your solve program, and how these phases can be used for phase extension and modification and so on. The methodology with which the SHELX suite identifies initial phases has also been discussed. This is purely based on the anomalous scattering, which will be pronounced at a wavelength where the anomalous part correction F double prime is more. And this could be done usually when you take your selenium methionine derivatized protein to the synchrotron and you collect the wavelength at a value where you have the absorption edge of selenium nearby. Similar pronounced anomalous scattering can also be done using lab source data collection by soaking with heavy atoms like iodine, eurobium, cerium, cesium and I3C, which will not disturb the overall structure. The PHENIX automated software for the structure determination is also explained in this module. Thank you.